Thank you. It is really my privilege to be a part of this anniversary celebration of such an esteemed organization. It's been fascinating to sit and listen to the rich and important history that this organization has had. And in the next few minutes, what I hope to share with you is the perspective from the Food and Drug Administration view of how critical animal research, but also how we also participate in things like 3Rs and ICFAM and so on to try to minimize use of animals. But at the same time, the use of animals is really a critical and important part for us to fulfill our regulatory mission to protect and promote both human and animal health. Next, oh, I guess I have this, right? Okay. So just the boilerplate disclaimer, although my comments were also very well informed by input from a number of FDA colleagues, including 2 veterinarians in the audience who are very actively engaged in our FDA vivarium programs. I'm going to try to hit on 3 major themes. The first is how important well-designed, scientifically valid studies in animals are to our regulatory decisions, how FDA supports application of the 3Rs principles, and our views on future trends in the use of animals and alternatives. So on the first theme, so I think I don't need to tell this audience how critically important animal models of human disease are for discovery, development, and evaluation of novel products. And in general, for FDA, animal studies are required for proof of concept and toxicity evaluation of regulated products. This includes not just medical products, but also things like food additives, food contact substances, sometimes contaminants of foods such as melamine, and things like PK, PD studies are really critical to inform dosing as we approach human clinical trials. And in some instances, such as medical countermeasures, efficacy studies cannot ethically be performed in humans, or they may not be feasible to perform in humans because of the rare natural incidence of certain diseases. And as I'll mention in a few minutes in more detail, they will require use of animal models. So FDA research, both our intramural research program as well as research that's done to support regulated products, obviously follows all of the animal welfare laws in addition to our own good laboratory practice regulations in the case of regulated studies that are submitted to the agency for review. But we also, if they're conducted using a regulated species, follow the Animal Welfare Act and those regulations. And if funded by the U.S. government, obviously must comply with the Public Health Service policy on humane care and use of animals. So I mentioned a few moments ago the animal rule in terms of requirements for animal efficacy studies. So what we're talking about here are things like agents that would, interventions to address agents of terrorism, chemical, biological, radiological, or sometimes things like antisera to snake venoms or mushroom poisoning and things like that, where you may not have a high incidence of the natural disease or activity, and therefore it's not feasible to do an efficacy study. And in the case of, for example, a vaccine where you really want to look at challenge, it's not ethical to do such a study. So in that case, we actually have modified the Code of Federal Regulations, Parts 314 and 601, which relate to new drug and biological drug products, and talk about what evidence would be required. And it would require a study in at least one animal species that is shown to have a reasonably well understood pathophysiological mechanism that's comparable to the human disease, and that you can design a study that has an animal study endpoint relevant to the desired benefit in humans, and that you can perform PK and PD studies in order to allow selection of an effective human dose. I want to talk for a moment about our intramural program. We have an active intramural research program that uses a variety of animal models to support the regulatory science needs of the agency. At Center for Vet Medicine and Center for Food Science and Nutrition, they actually have a nice facility that supports a variety of large animal research models, including cows for milk and meat safety studies, a very large aquaculture research program, and I'll talk about that in a little bit more detail. Use of poultry to study mechanisms of antimicrobial resistance, as well as how to evaluate detection of antimicrobial resistance. They have pigs that are used by colleagues colleagues at Center for Devices and Radiological Health as models for cardiovascular disease, and they also have goats in order to support biomarker research. As I mentioned, they have a very robust aquaculture research program. I'm not going to name all the species, but you can see it's fairly varied and diverse. And the goals are to facilitate new drug evaluation for aquatic species while also assisting with the agency's surveillance efforts to protect the food supply from illegal drugs or chemicals contaminating fish that are used in our food. I'm not going to read through the studies, but you can see it really, there's a wide variety of studies that are, again, in support of our regulatory mission. In the biologics and drugs world, our regulatory science programs also use a variety of different animal models, everything from Drosophila for high-throughput genetic screens, Xenopus laevis for developmental biology studies, mice and other rodents like rats and guinea pigs as disease models for various infectious agents that we may be evaluating in terms of vaccines or therapeutics. And then we have a very active influenza preparedness program to participate in developing reagents for lot release testing for both seasonal and pandemic influenza virus. And to support that, we use sheep to develop antisera, which are actually distributed worldwide to all manufacturers of influenza vaccines to calibrate their potency reagents. And then we also use things like ferrets as disease models for influenza as well to look at vaccine seed strains and make sure they are no longer pathogenic. And in rare instances, we also still use non-human primates. They sometimes provide unique models that can't be served by other species for certain diseases, and again are important for models of infectious agents and vaccine studies. And I wanted to just highlight one recent example, which is a new model that's been developed by Todd Merkel in our center to address an emerging public health need, which is this resurgence of whooping cough caused by Bordetella pertussis. As you may know, since really 2010, there's been an increasing trend, and the 2012 CDC data is showing approximately 50,000 cases in the U.S. So this is really a new and emerging and important critical public health issue. So obviously this is happening in people who got the vaccine, but the vaccine doesn't have long-term effectiveness, and that's why we're seeing these outbreaks. And so this is really driving a need in the community to develop new vaccines and also to have a better model to look at why these vaccines are no longer effective. We actually don't even fully understand what are the immune correlates of protection for pertussis. And so Todd Merkel has developed a baboon model which replaces what used to be done in rhesus macaque and has shown that actually it's a more robust model, more reproducible to the human disease, showing airborne transmission through the respiratory mucosa and so on. And so this is really an important development to help facilitate further vaccine development. So the second theme, the 3 R's, which everybody has mentioned in their talks, because how can we not mention this? This is critically important. While we recognize the importance of using animals, we also, as everybody has says, want to minimize and use them wisely and look for alternatives. We actively participate in things like ICVAM, Tox 21 was mentioned earlier, to look at validation and adoption of alternative approaches. We are investigating in vitro approaches for toxicology screens, such as organs on a chip and other methods, such as stem cell derived cell lines that could be used for toxicology screens, looking at replacement with lower phylogenetic species, looking at how to develop a research protocol that uses earlier endpoints to reduce suffering when possible. In addition, we also refine the use of animals by having active enrichment programs. These are just some pictures of a few from our large animal program, showing things like co-housing, paired housing of non-human primates to promote socialization, but we also have an active enrichment program for our small animals as well. ICVAM, I think you all know, is Interagency Coordinating Committee on the Validation of Alternative Methods. It's represented by 15 different government agencies, including the FDA, and its role is to facilitate and promote development, validation, and regulatory acceptance of new and revised regulatory test methods. Really, its mission is the 3 R's. And so we we participate very actively in this, along with the NICEDM, which provides the framework for providing the translation of the basic science into the validation, evaluation, and regulatory acceptance of these new tools. I want to give one example of some work that we have promoted, which is replacement of the poliovirus monkey neurovirulence test. I thought it was kind of fortuitous, actually, that in 1953 that was the poliovirus vaccine, because I added that as an example. And as many of you may know, when the live poliovirus vaccine was introduced, there were cases of poliovirus that were induced, because although the live poliovirus was attenuated, it had the occasion to revert back to its neurovirulent phenotype. So in response to that, the regulatory agencies, and actually it was an internationally accepted standard, developed this, what was called the monkey neurovirulence test. And it actually required that each lot be tested twice in non-human primates, first by a manufacturer and then by the National Control Authority. In our case, the FDA, Center for Biologics, performed this assay. And so in the end, each lot required 200 monkeys, or almost 200 monkeys, to release one trivalent bulk. So in the late 1980s, actually, the receptor for poliovirus was identified, and this led to the development of a transgenic mouse model. And we actually at CBER adapted this transgenic mouse model by identifying a way to do intraspinal inoculation, and allowed us to develop very sensitive neurological endpoints for looking at the neurovirulence test. And in the 1990s, a large collaborative study led by the FDA as well as the World Health Organization led to adoption by 1999 and 2000 of this as an alternative to the monkey neurovirulence test. So this actually should say poliovirus mouse neurovirulence test. I apologize for that. Finally, future trends. Telemetry is a critical part of allowing more precise in-life measurements in small animals and large animals. Non-invasive imaging in order to use fewer animals as well. Both of these, as we can gain more from in-life measurements, will obviously reduce the total number of animals used in any particular study. For us, having more predictive models that better recapitulate human disease and the human response is going to be really important for improved evaluation of products. And I think also as we understand more mechanistic properties, that may also support developing alternative approaches such as cell-based alternatives that get us away from using animals in some cases. And so we hope going forward that we'll have improved in vitro and in silico approaches to make evaluations. Just a few quick examples from our own laboratories. Todd Merkel, again, has developed a very nice model. In this case, anthrax was modified to contain the gene for luciferase. This allows, using in vitro imaging system in the presence of luciferin, very precise staging of anthrax infection. And this is actually a really important way, again, to refine the use of animals, because this allows for more precise staging for post-exposure prophylaxis or therapeutic testing. We also are looking at a chimeric mouse model as an alternative to the chimpanzee for propagation and evaluation of hepatitis C virus and vaccines. In this case, it's using a urokinase protease that is under the control of a promoter that allows for this gene to be expressed relatively late in development. This allows mice to survive later, but eventually they develop damaged livers, which can then be replaced by human hepatocytes. And we've been evaluating this model for its ability to propagate hepatitis C virus. And this is just showing that we can recapitulate the features of the model, showing that the mouse is engrafted with human liver and that we can detect hepatitis C virus in those engrafted mice but not control mice. So I'll just finish with where I started, which is that research in animals is really a critical component required for FDA's regulatory science program, while also being absolutely important for regulatory decision making and fulfilling our mission to protect and promote human and animal health. But of course, in those cases where animals are used, it always needs to be done in a manner that's humane, well-designed, and controlled studies, so that when we're using animals in research, to support regulatory decision. It's done thoughtfully and carefully. And I finally just want to finish with a thanks. Even though it's a small font, it's a huge thanks to a number of my colleagues who provided input into this presentation. Dr. David White in Office of Foods and Vet Medicine, Center for Biologics colleagues John Dennis, Constantine Chumakov, and Marion Major, and in the Office of Chief Scientist, our own Chief Scientist, Jesse Goodman, and Pamela Chamberlain, who's here. And I thank you for your attention.